All right, so all blood cells originate from the red bone marrow, which is in adults. It's a little bit different in children, but in adults, the red bone marrow is found in flat and irregular bones. So ribs, vertebrae, sternum, pelvis. Um, a lot of times when you do bone marrow transplants, they do it in the ilium along the iliac crest. That's the and when they do a biopsy, that's where they get it from. So the various blood cells develop from a single stem cell. Those stem cells up at the top of this chart are called the pluripotential hematopoietic stem cells. So during the process of hematopoiesis, which is also called hemopoiesis, the differentiation process they're differentiated into 5 different types of committed stem cells, meaning they're going to develop, like if you have one type, it's going to develop into that line of blood cells. What am I trying to say here? So like the ones that are going to develop into red blood cells are going to develop into red blood cells. You're not going to get that stem cell type and have it develop into thrombocytes. Okay, so hopefully that makes sense. So the cells are they're going to mature and differentiate into the different functional cells that are needed by the body. Any pathological condition of blood that is involving the cellular component is called a dyscrasia. So anemia is a dyscrasia, leukocytopenia is a dyscrasia. So we're going to talk about all those individual disorders. So anything having to do with the cellular component of blood is a dyscrasia, any disorder. So I know that on this, you can't really see the little individual details of the chart, but on page 192 in the text, you can take a look at this chart and see the details of it. I think it's pretty cool how they develop into things. So the individual cells, erythrocytes, red blood cells, they're biconcave flexible discs. Sometimes you'll see, like in the book, it says they're like donuts, but they're not really like donuts. They're like bialys. So they're not like bagels where there's a hole all the way through, or donuts where there's a hole all the way through. It's like a bialy where it's round on the side and flat in the middle. So it doesn't go all the way through like a donut or a bagel. So that's what they mean by biconcave. They have to be flexible so they can fit through those itty bitty capillaries. There is no nucleus in the mature state. They do contain hemoglobin. So there's the globin portion, and there's the heme group, which the iron binds to, or the oxygen binds to. Sorry, the iron is part of the heme group. They have the lifespan of about 120 days. When the body decides it needs more red blood cells, the kidney produces erythropoietin, which is a hormone. It stimulates erythrocyte production in the red bone marrow. And the trigger that makes the kidney decide to do that is hypoxia. So if you have insufficient oxygen available to cells, then your kidney says, "Oh, we need more red blood cells. Let's release some erythropoietin," and that stimulates red blood cell production in the bone marrow. So if you watch the Tour de France or any kind of international event, one of the things they test the athletes for is what they call blood doping. And what they mean by blood doping is that erythropoietin is artificially injected into the athlete to produce more red blood cells to make them have better oxygen, better VO2, and they can work harder. So that's a way of cheating, basically. Although there's a long tradition of cheating in the Tour de France, so I'm not going to say one thing or another about it, but that's how they do it. They use an external source of erythropoietin to create new red blood cells. And there are other ways to do it to try to evade testing, just to make it interesting. So normally red blood cells, there are a lot of them. They're like millions. The red blood cells constitute most of the cell volume in blood. So your hematocrit measures the cell volume, and most of that is red blood cells with a few white blood cells thrown in. So like when you look at these micrographs, the top picture on here is an electron micrograph. So you see the cells really up close. The other one is a light microscope micrograph. And you can see the little iron colored ones, those are the red blood cells. The big ones with nuclei, those are the leukocytes, the white blood cells. And the tiny little purple ones, those are the platelets. And the reason they're purple is because they take up the stain that they use on a blood smear. So hemoglobin is the main reason for red blood cells to bind to the oxygen and take it out to the cells. So deoxygenated hemoglobin is sort of bluish red, and regular hemoglobin is bright red. And that shows you the difference in color between veins and arteries. Like arterial blood is bright red, venous blood is blue. So if you have visible veins on your hand, I do. Not everybody does. I guess I have pale skin or something, but you can see my veins, they're blue. I'll just tell you, my veins are blue. So that bluish red color is found in venous blood. Of course, if you cut yourself accidentally or draw blood, as soon as the venous blood is exposed to oxygen in the air, it turns more red. So most of the CO2 in blood is not carried in hemoglobin. Most of it's carried in the buffer system. Remember when we talked about that? acid-base metabolism and all that. So most of it is carried in the blood as bicarbonate ion, so it can participate in that buffer system. Bicarbonate ion is also created by the kidneys. So the kidneys are so important in everything. Kidneys are big players in the circulatory system. Kidneys are big players in a lot of stuff. They're the big fish. They got their hands in everything. You can't live without kidneys, but you can't live without red blood cells either. So they're little, important little guys. So leukocytes, I don't have a slide for leukocytes. They're important though. There aren't as many of them as erythrocytes, as you can see in this blood smear. They are only about 1% of the blood volume. They are super important though. All of them differentiate from their stem cells in the bone marrow, and then they go off to do their jobs. So the T lymphocytes go for training to the thymus, and the B lymphocytes are trained in the bone marrow, and then they get to do their jobs. So once they're developed, then they get out into the bloodstream and they can participate in the inflammatory response and the immune responses. They can do all their stuff. So sometimes you see like different types of leukocytes in blood smears, some with big nuclei, some with lobed nuclei, and that's how they tell them apart just visually. But lymphocytes make up about 30 to 40% of white blood cells, of leukocytes. T and B lymphocytes in the immune response, we talked about a lot last quarter, and they're big players in the immune system. Neutrophils are the most common leukocyte, about 50 to 60% of white blood cells, but they only survive 4 days. So they go out, they phagocytize things, and then they die, and you get new ones. Hopefully you get new ones. If you don't get new ones, then you have neutropenia, never good. They're the first to respond to any tissue damage and start phagocytosis. So we got to have them. It's really important. You see increased neutrophils with a bacterial infection. A lot of times they'll do a complete blood count, counting all the cells, and then they do a differential count, differentiating the different types of white blood cells so you can tell what's happening. So basophils become mast cells that release histamine and heparin. They might be just wandering around, or they might be hanging out in lymph nodes, or they might be attached to particular tissues. So basophils are sort of all over the place. Eosinophils tend to fight against the effect of histamine. They are responding to an allergic response. They're increased when you have allergic reactions and parasitic infections. Monocytes enter the tissues and become macrophages, which act as phagocytes. And as you know, they're really important in healing and in the immune response. So when you get the differential count, it tells you the proportion of each of those different types of white blood cells. The thrombocytes are also called platelets. They're a big part of the clotting process or hemostasis. So hemostasis is important to keep you from bleeding too much. Thrombocytes aren't really cells. They're kind of small non-nucleated fragments that broke up from large, they're called the megakaryocytes that break up into platelets. Platelets stick to damaged tissue as well as to each other, and they form a platelet plug that seals small breaks in blood vessels. Or they can adhere to rough surfaces and foreign material. So that's why we'll start talking about atherosclerosis, the inside of the artery gets all icky and rough, and you form more clots, and that's a problem. That's how you end up having a heart attack. So we don't want to do that. Sometimes one of the treatments for a common drug treatment for blood clotting is aspirin, acetylsalicylic acid. It reduces platelet adhesion, and of course that means you can have an increased tendency to bleed. Thrombocytes can initiate the clotting process. So if you have an injury first, the blood vessel is going to constrict. There's a little bit of a vascular spasm. In small blood vessels, that decreases the blood flow and may allow a platelet plug to form. So you get that platelet clot. The thrombocytes adhere to the underlying tissue at the site of injury, and if the blood vessel's small, the platelet plug can actually kind of stop the bleeding right there. After that, then the coagulation mechanism, which is required in larger vessels, takes effect. So in larger vessels, you can't plug it all with just a platelet plug. There has to be more that goes on. So a lot of the clotting factors are present in inactive form in the circulation. And once they're activated, they go through a series of reactions that help clot the blood. So I don't expect you to know the whole sequence of hemostasis, but basically it's a cascade of events and they're different factors. So if an individual factor is missing, it stops the clotting. And so in blood clotting disorders or hemophilias, there is something wrong with one of the factors. So hemophilia A, which is the genetic form of hemophilia, factor 8 is the one that's involved. And so generally, damaged tissue and platelets release factors that stimulate a series of reactions and produce prothrombin activator. Prothrombin activator is also called factor 2. It's usually inactive in the plasma and it's converted into thrombin. Thrombin is a molecule that acts as a procoagulant and an anticoagulant, interestingly, depending on which reaction it's involved in. Fibrinogen, which is known as factor 1, and the factors aren't in order, just to make it interesting. They're in the order that they were discovered in. So you can't memorize the sequence by memorizing the order of the numbers, which is darned annoying, you know. But fibrinogen is converted into fibrin threads by being activated by the molecule thrombin, and that forms a mesh which traps cells and makes up a solid clot or a thrombus, stopping the flow of blood. So you want clots sometimes and you don't want clots sometimes. So there's a fine balance between clotting and not clotting. And you got to have the right balance or it just doesn't work. Eventually, ideally, and this is how it's supposed to work, the clot shrinks or retracts and it pulls the edges of the damaged tissue closer and seals the site. And then vitamin K is involved in the synthesis of clotting factors, so it's important. and calcium ions are involved as well. After that, you have to, once you've sealed the site and prevented blood loss, then you have to get rid of those clots. So there's actually a system that dissolves, it's called fibrinolysis, that dissolves the clot. So you have to build the clot, you have to seal the site, you have to fix things, and then you have to break down the clot. So if any of that doesn't work right, you're in trouble. So if you have a clot wandering around in your circulatory system, that could cause a heart attack, that could cause a stroke, that could cause a pulmonary embolism. So lots of issues with clots. So you have to have just the right amount of clotting and not too much. Just like a lot of things in the body. It's very Goldilocks. It can't be too much, can't be too little. It has to be just right. So we're not going to really go into detail about the clotting, but just know that if you have people who are on blood thinners, any types of blood thinners, whether it's aspirin, usually on aspirin, they don't require you to go in and get regular blood tests. There's another prescription blood thinner that you don't have to get regular tests. They're warfarin or Coumadin. You do have to get regular tests, and we'll talk a little bit about blood testing right towards the end. Blood typing is based on antigens in the plasma membrane of erythrocytes. So if you're type O blood, you have no antigens, and you have antibodies against A and B. You can only receive a transfusion from another O. Luckily, lots of people have O's, so don't worry if you're type O. If you are a type A, you have A antigens, but you have anti-B antibodies. So you can receive a transfusion from O. Anybody can receive a transfusion from O, or from A. If you're a B, you have B antigens and anti-A antibodies. So you can receive a transfusion from O or B, but not A and not AB. And if you're AB, you have A and B antigens, no antibodies, and you can receive a transfusion from anybody because you have no antibodies. So O's are considered the universal donor. They can give blood to anybody because they have no antigens. ABs are considered the universal recipient. They can receive blood from anybody. And A's and B's are just A's and B's, just normal. So it's kind of it. I think it's really interesting, but there's also something that's called the RH factor. If you have it, you're RH positive. If you don't have it, you're RH negative. If you have an incompatibility in RH factor between parents, that can cause problems at birth. So you usually, when you get your blood type, it'll be like A positive or O positive or B positive or O negative, and that means your type and your RH factor. So there are certain sports where you're required to have your blood type embroidered on your clothes, just in case you're in an accident, you have to get a transfusion. But auto racing is one of those sports. Just a fun fact. So you know, it's very interesting that blood typing is interesting. And if you get a transfusion from someone that you've got antibodies to their type of blood, you're going to have that type 2 hypersensitivity reaction, that hemolytic reaction that's going to break down your red blood cells. So it's pretty important for the blood to be typed before you donate it to somebody else. A lot of people, before they have surgery, they have a planned surgery, they will go and donate blood ahead of time and get their own blood back if they need blood. And that way, you know, you're not going to have a reaction because it's your blood. So diagnostically, a complete blood count, CBC, is a really common test. It counts everything: total red blood cells, white blood cells, and platelets. They do it from a smear. Leukocytosis is where you have too many white blood cells, and it's associated with inflammation or infection. Leukopenia is decreased white blood cells, and it's associated with some viral infections, and radiation and chemotherapy also. They usually closely watch your blood counts when you're getting chemo and radiation, just to make sure that everything's going okay. Increased eosinophils are common in allergic reactions. To tell the difference between the different blood White blood cells, they do a differential count. The morphology is observed with blood smears. So they smear the blood on a slide and look at it under the microscope, and you look at the size, shape, uniformity, and maturity of cells. Different types of anemia can be distinguished with a morphology test. Hematocrit is the percent by volume of cellular elements in the blood, and it's expressed as a percentage. So it'll say your hematocrit is 42% or something like that. If your hematocrit is low, you're not getting enough oxygen, you might be dizzy or fatigued. Hemoglobin is the amount of hemoglobin per unit volume of blood, so it's expressed that way as amount per unit volume. Mean corpuscular volume indicates the oxygen carrying capacity of blood, or MCV. They used to call blood cells corpuscles back in the, I don't know, the 1800s, a long time ago. Now we know they're cells. This was before they knew about cells. Reticulocyte count is an assessment of bone marrow function. Chemical analysis determines the serum levels of lots of different chemicals that are in the serum. Iron, B12, cholesterol, all that stuff. So glucose. So a lot of the chemical analysis that they do on blood is determining the level of those different areas. If you're having to get clot, if you're having clotting or bleeding issues, one or the other, you need to do bleeding time to measure platelet function, or prothrombin time is another different test. They measure the function of various factors in the coagulation process. And an international normalized ratio, or INR, is a standard version of a prothrombin test. So if someone's on Coumadin or warfarin, they are going to have to go in and get an INR pretty frequently to make sure they're not too uncoagulated and they're not too coagulated. They have to be in that just right category. If you get too much coagulation, you get blood clots. You get not enough, you get bleeding. So you have to be right in between. So you can get blood transfusions, obviously, as whole blood, or packed red blood cells, or packed platelets. If somebody has severe anemia or thrombocytopenia, they're often given those blood products. Plasma or colloidal volume expanding solutions are administered. They don't have any cells in them, so you're not going to have a reaction to them because they don't have the antigens or antibodies. Those are carried on cells. A lot of times when you have issues with osmotic and hydrostatic pressures, they give you plasma or volume expanding solutions. Artificial blood products are available, but there's not a single one of them that does all of the functions of normal whole blood, which I think is interesting. They are compatible with all blood types, which is pretty handy. There are some that are made from human blood, so they're artificially made. There are some of them that are just not blood products. There's something else. There are lots of different things always being developed, and they have to be approved by the FDA before they can be used. There are some recombinant DNA products that have been tested and approved to replace erythropoietin, and sometimes they'll use it for people with anemia related to cancer or chronic renal failure. Anytime you get a blood transfusion, there is a risk of an immune reaction or infection. And so by using these artificial products, you are reducing the risk of infection. So that's the whole point of them. There's also bone marrow or stem cell transplantation. You need a close tissue match. It's used to treat some cancers or severe immune deficiency or severe blood cell diseases. So your human leukocyte antigen, or your HLA type, has to closely match the donors. They extract marrow stem cells from the donor's pelvic bone, and they filter it and they infuse it into the recipient's vein. So then that lets you grow new bone marrow, basically. And they expect normal cells to appear in several weeks. And usually, if you have like a cancer or blood disease, they do chemotherapy or radiation to destroy the abnormal cells, destroy the tumor cells, and then they transplant the new cells. So in that time when you're growing your new bone marrow, you are extremely immune compromised, and you need to take huge precautions to keep from getting sick. So drug treatments are used to replace blood clotting factors in people that don't have them. There are lots of different medications out now that treat things, deficiencies in the clotting process. So it's an interesting area that we're not really going to go that much into, but just to know that there are drugs to treat that.